All right, recording. Nice. Well, folks, all right. So starting out, Brian, I think he's going to take control, and we're going to start our microscopy adventure with what you're saying. And just Keisha and Brian, is there any kind of like orientation tips kind of before we get started? Like where did you what did you do to get to this point where you're viewing the sample?
Yeah, I think you're going to start us off, right? With that.
Yeah.
Yeah.
I am. And I'll orient you to my space right now. I'm actually sitting on a floor. So Ollie, for the first time ever, has the ability to be present for webinars. And it seems like
Hi,
he really
Ollie.
wants to be here. So it's gonna be a little weird. But yeah, I want to talk about qualitative assessments, right? So we do a lot of different work. You guys probably know us for the windrows we build, and we've been in this work for a long time, sharing with folks. We went to Soil Foodweb school, we're a certified lab. So in the beginning of our career, we really focused on quantitative assessments, which they're still qualitative because it's a guess, but we're using much more specific numbers. But you know what? It takes a lot of time to perform these samples. They're a little bit expensive. And as we've worked through time, we've realized that a qualitative sample, just looking at, no kisses right now, looking at our samples and just telling a story is often enough. And a lot of the times when we're working with our clients, like these farmers don't necessarily have the time to go through a long schooling program. They don't really want to learn how to be consultants or how to be a lab. And so what we've learned is just in a few days' time, we can sit down, do some microbe adventures, talk about, who we're seeing, what the morphology of these organisms are, kind of like what we're going to do today. We're going to walk you through the samples and just talk about what we're seeing. And then we can have a dialogue with our clients. They know how to make a sample, they know how to use their microscopes. And from a great distance, I can talk to them about their compost and what it's looking like under the microscope. So, for example, we have a client in Florida who has a large scale lot that we built for her. And she was concerned about her microbes the other day. She called and said, I don't know if they're looking as good as they did when you first built them. So she got on the phone with us while she was looking at a sample and she said, every time I move my microscope, I'm seeing 3 or 4 testate amoeba. I'm seeing about 2 to 6 nematodes per drop. And there's actually fungi everywhere, lots of different colors of it, and it's branching out all over the place. So that right there is enough of a story for me to say, hey, you know what? It sounds like that compost is still a thriving ecosystem and no problem to use it. So today we're going to go around and just kind of see what that looks like, qualitative instead of quantitative. Brian?
Yeah. I mean, my thoughts very much mirror yours because in the work that we do, I do a lot of quantitative analysis for some of my clients because they are more in that kind of hardcore data-driven space, and it works well for them. But I have other clients that are like, you know what? Again, like you mentioned the affordability of it, and plus they want to be able to go look at their soils, look at their compost, look at their environment themselves because I start to share with them the microscope. They get a microscope, but it's really just kind of breaking down the barriers for them so that they have the ability just to look at it and make their own impressions. And that was, when we talked about putting a class like this together, one of the things that we really wanted to kind of focus in on is how do we make microscopes much more accessible to people? Because when you start talking about quantitative and you're talking about, measuring lengths of fungal hyphae and diameter and getting the counts and all the math that goes associated with it, that really either, turns people off or they get, kind of scared by that. And so that they don't want to use their microscope. And we want to take an approach that says, no, we want you to have a good time looking at the microscope, but let's just focus on the skills of how to use it. Just so you know how to make proper dilutions, you know how to be able to put a drop on a slide, and you know how to be able to scan through a sample. And then you start to build an intuition. And I'll tell you, it's kind of funny after seeing thousands and thousands and thousands of samples at this point now in my career, you build a strong intuition. I can take a look at a drop of something and I can start to make some pretty good impressions about the quality of the microbial community. Is there soil aggregation that's there? And it's just simply because I built an intuition over time. And that's just a lot of hours of eyeballs on the microscope. And anybody can really gain that intuition without even having to do quantitative at all. And so, our focus on our course is how do we make this so that you can do qualitative assessments pretty easy, but then also how do you make it fun too? doing like a microbe adventure. In today's talk, I decided to do a microbe adventure of my yard. So I went out and pulled 5 different samples. One from a garden bed, one from a highly disturbed soil that I haven't really done anything with yet, vermicompost, a wood chip pile, and I'm doing an experiment making fish hydrolysate. So these are pretty wildly different, samples. And for me, it's going to give me some feedback about how are some of the things that I'm doing in my yard, how are they doing? And also sometimes it's just cool. Sometimes you'll see stuff. You're like, oh wow, I haven't seen something like that before. And I also live in a very lucky environment where I got some old growth forest by me. I've got some wetlands. And sometimes it's just fun to go out there and start pulling samples from these areas and just look, even though I'm not going to be doing any kind of management in that space, it's still there's some really neat things out there that I could actually see. So yeah, I'm excited actually to be able to share this. And I'm excited for us to be able to run this course to kind of teach people this, philosophy and mindset, but then also the tools and techniques in order to be able to use a microscope effectively. Casey.
Oh, hey, hey, hey. Sorry, I had to run out for a second there, move my car. I was blocking someone. But yeah, no, this is really cool. I'm excited that we're putting together something like Brian said, like it's a little simpler, it's a little more teaching people how to do something so they're not so intimidated by it. I remember back in the day when we first learned how to do assessments and things, and we were doing quantitative assessments, we had to like write everything out by hand. We used to have like little pieces sheets of paper, and then you actually had to sit there and do all the math, and it just confused people and scared people and made it seem like so intense and so over the top. And yeah, I don't know. I like the idea of teaching this idea, like the way to do it where yeah, you're exploring, you're looking, you're on an adventure through that soil, and you can start to pick up and glean like really good information throughout a soil sample without having to write down any numbers or calculate anything. It's more of a mental calculation because it can be to where, you look at some samples and it's just, you notice right away. It's just like little teeny dots of sand and there's nothing else there. Or you throw another sample up on and you see these dark browns, you see these ambers, you see little bits of soil clumped together, you see testate amema or a nematode. These are like little signs that we can use to just see how things are progressing. And yeah, I don't know. I'm really excited for this course just because it demystifies it, right? It gets people comfortable with it. Like everybody should be sticking their nose in a microscope and just having fun. It shouldn't be a stressful thing. So,
Yeah.
yeah.
And I just found a really nice juicy strand of fungi. We didn't wait. I was like, oh, I found one. Hooray.
But yeah, well, go ahead, Raleigh, take it.
Yeah.
Anyway, well, the what part I'm excited about, yeah, I think qualitative analysis, it's just the usefulness of it. It's just the point where it's like, because I know like I spent a lot of time in the lab. I of course did Soil Foodweb school and did the microscopy program, and it's like you're doing the whole SMAP and quantitative analysis, and even after doing that like 15, 20 times, 30 times, it still takes so long. If I'm just looking at compost quality, I'd rather like just I want to do the scan. I want to do this more rapidly and not have to spend hours and hours doing this to get like a number. And sometimes the number can be wildly different, even if you're doing the same scan or if someone else doing it. It's like that's the part that's pretty wild. But I don't know. I'm big excited. But without further ado, how about let's get started with this
Sure.
exploration.
All right. Do you want me to? You have a fungi right there, Raleigh.
Okay,
You want to
how
bring
about
that up?
Raleigh really
I'll
wants to share his
I
fun fact.
feel like,
Yeah.
Raleigh.
all right.
Um,
Well,
And,
all
well,
wait,
right.
and Raleigh, tell us
everybody, the
about the
awesome
sample.
name of your
So
compost pile.
this is a compost that's been sitting in the backyard. We made this in April. This is from my Sargent compost pile. And it actually didn't make it past the third turn. So I was like, dang it. It didn't make it up to regs, but it sat there, what's that? May, June, July, August, September, October. Wow, almost 7 months. So it's been sitting here, just aging. I've been keeping it watered. And the good news is like looking under a microscope now, I'm seeing that like there's tons of aggregates, and I see, surprisingly lots of fungi, which is great. And this is at 1 to 10 dilution. The objective is, I think, what, 40x here? Yeah, 40x. And just, I wanted to show you guys, like, because I did that video where I looked at Miracle Grow and the community compost, and this at 40x, there was nothing. The commercial compost just had bacteria, not a single fungal strand, no nematodes. And meanwhile, I'm looking at this sample of the
Hey, Raleigh,
backyard yeah,
can I interrupt you just for a second just
please.
to throw a little techno thing out there? So when Raleigh says he's using his 40x objective, for those of you that don't know, that's the power magnification of the objective, but his eyepieces also add another 10x. So his total magnification in this case is 400 times. So I just wanted to throw that out there.
Yeah. And because can
Drop a
everybody,
little knowledge as we go along.
I don't know, knowledge. Can everybody see the screen pretty well?
Oh yeah,
Yeah,
Okay,
that's
that's
good.
sweet.
perfect.
Well, yeah, I just love, like here it's like, oh, big, healthy fungal strand, a nice spore here.
Yeah.
And I'm sure if I focus more through these aggregates, I can see, more fungi are hiding in the aggregates. So
And
it's
actually,
just
if you get that fungal strand into focus, you can really see the septa on that one, the little
yeah,
dividing lines down the middle of it.
I know they're so
Is
clear. It's
real.
super nice.
That's, just a great indicator of healthy, non-pathogenic fungi. It's investing a lot of energy into its body, into its own structure because it's free living.
Hmm.
Its plan is to not get out there and jump inside of something else and feed off of it. So it has to spend energy investing in itself. And that's where you're getting that dark color. You're getting those nice septated walls, those little dash black lines down through the middle of that fungi. So
Yeah.
yeah, very cool. That's a nice one.
And this, I'm using an Omax scope. This was like probably 800 when I bought it all the way back in like 2020. So I'm excited to see like Brian's scope, which is a sweet LW one, seeing the things that we see. Well, this
No,
was
to be
this.
honest with you, Raleigh, I mean,
Uh,
you're getting great optics off of this. I mean, that's the whole thing too, is people will be like, oh, do I have to go spend thousands and thousands of dollars on a microscope? And the answer is really no. I mean, it depends on how much you use it. Like for myself and Keisha and Casey, where can spend sometimes 8 hours a day under the microscope. Having really good sharp optics helps with eye
yeah,
fatigue and those kind of things. But if you're just going to be going around casually every so often, you can get away with a fairly inexpensive microscope and still get wonderful imagery.
Yeah, that's
yeah,
right.
for sure.
It kind of actually makes me think of to show off one little thing here that like is different like with Raleigh's microscope and Brian's is if everybody looks at the bottom edge, Raleigh, don't move real quick.
Oh, sorry.
Hold it in focus. If you go back over to your fungi, put it in view real quick, and you'll see like what changes like the differentiations in the microscopes is right there is fine. You can leave it right there. If you look at the
Mhm.
bottom of the fungi near that spore down there, you can see how it kind of starts to bleed out of focus a little bit and like all the perimeter. That's really the big difference that you get with like a fancier microscope. It's the fancier objective lenses. The objective lenses are flat. The more expensive they get, the flatter they are. So as you have the cheaper ones, really what you're losing is about that, just that outer little edge of that vision on your microscope or when you're looking at your sample through your eyepieces is going to be a little bit blurry because the glass curves just ever so slightly. So really that's the biggest difference that you see with the more expensive you get up the ladder is just like you get from 70% to 80% to 90% to maybe 99% of your field of view is perfectly in focus because the glass is perfectly flat.
God, how do we explain the effect that your eyes get with the expensive microscopes? The lighting, maybe?
Yeah.
Yeah, the
Yeah.
Do you know
Yeah.
what
ink
I
on
mean?
the
Like
eyes.
I like if
Yeah.
Yeah.
I sit like I think I did a study with Elaine one time where we were looking at compost tea. And I think Brian, like you're the one that decided how many replications we needed to do. Like you guys were monsters. We were literally doing like 30 samples a day. And it was like the difference between the LW and the Omax was like I
Yeah.
like literally I think it might be the lighting, the difference between
Yeah.
LED and halogen.
Halogen.
And
Yeah,
halogen lights, yeah. And
halogen.
the sharpness of focus too. Just I think for me it's just the eye fatigue goes down with the better optics that you have. And I'll tell you, the objective lenses, you can buy objective lenses that will cost much more than the microscope itself. I mean, there are
Mhm.
objective lenses out there that cost in the multiples of thousands of dollars for
Wow.
just one objective lens. Now, do you have to go that crazy? No, but if you're going to be doing something where if I was doing microscope work every single day, 8 hours a day, oh, you bet I would probably be looking a lot more and investing into really, really great optics that are out there. Um, so yeah, it does help for sure, but also don't make it a barrier for yourself too. Don't think you have to get something that's many thousands of dollars, you can, like I said, get away with something that's fairly cheap.
Yeah.
And like Casey was describing, the plan objectives, that's something that I'd be looking for too. Get something that's plan, not semi-plan, and that means it gets you the sharper all the way out to the edges. Just great. Feels like doesn't like there's a Where's Waldo moment in here when we start looking at what else do we see in the sample?
Oh wait, can you guys see my arrow?
Oh yeah.
Okay,
Yeah,
sweet. So yeah, I was trying to think like, oh, is that a testate amoeba? I'm trying to like focus through, and it doesn't quite look like it. It has the shape, but as I focus, it doesn't look like it.
it looks a little minerally to me,
Yeah, a little
a little
minerally.
shiny.
Let me focus through here a bit more. I'm trying to think. I mean, I see, of course, I assume these are spores.
It
Hmm.
is. Yeah.
Fungal,
Yeah,
that looks like a fungal spore to me. Yep.
fungal spore, definitely fungal spory. But in this field, I mean, I see this, some things are hiding in this aggregate. If I zoom through, like there's, I think is more fungi kind of like within this aggregate. Um,
That's a good thing to point out right now. If we were counting this, right, if we wanted to be accurate,
yeah,
have to assume a lot of things that like, the fungal hyphae is in fact fungal hyphae. It's really moving in a way where it's hard to focus on one point of on all of it at one time. So we would have to assume how long it was when it was inside the aggregate. But that tells a story, like it tells a really nice story. Like we're looking at possibly two different species, three different species of fungi. We're seeing it come in and outside of aggregates, which is telling a story of creating structure. Um, and yeah, you can see the fungal spores right there, which is, potential of
More.
more. As well, that fungal hyphae could be alive. We don't know if it is or not, but
mhm.
there's a lot of potential in just this one view. Raleigh, what's it look like if you just move somewhere else
Yeah,
with
for sure. I mean, this was a
looking?
less
Without looking.
aggregated chunk of the sample, but as I'm scanning around, I mean, of course, there's like,
Oh,
Boom. There we
oh,
go.
stop.
Stop.
ooh,
Go up.
here we go.
Bottom.
Is this
All
guy?
right. Right there. Stay.
Oh, two.
So now like this like gives us even more of a story. We've done two movements now with our
Mhm.
microscope randomly. And all of a sudden now we see another spore. We see more fungal hyphae. This one's a little bit different color. It's a little
Mhm.
bit
A little thinner.
less in diameter. It does have septa. We see that testate amoeba. And this is the second testate amoeba in Raleigh's sample that I think is insisting, which is making me a little bit curious about his moisture level, but only curious. Sometimes when the compost gets a little dry, you'll see the testate amoeba start to insist inside their test.
Yeah, do you see the testate amoeba there on the side, Raleigh?
This guy?
Yep.
Mhm.
And then you see how
Yeah.
there's actually a cyst inside, not
Interesting.
the little circle inside is a
Yeah.
cyst, not the amoeba, so it's gone dormant.
Yeah.
Oh,
And
that's
that's kind of cool because it's telling you like, hey, the testate amoeba, the environment you put them in, it was gentle enough that they were able to naturally insist and do the thing that they do that protects them. So
yeah.
that's a little bit of story right there. It's not, this isn't incredible, strong data, but it's a lot like walking through the woods and observing the life around us and trying to put that all together.
I think what's interesting is looking at this, it's like, yeah, I do the squeeze test, and even though I'm at that 50% moisture where it's like, oh, that one drop of water runs out and it's raining outside, it's still been under a tarp for a long time. So I might let it get more moisture if that's the sign that it's like, okay, if this cyst is dormant, which means it's not enough moisture, then I need to get more moisture into this pile.
And we did have a question coming from Sophia
Yeah,
um, around, not a vacuole. Um, and somebody also says they're not a spore that amoeba ate. Um, so on the left-hand side. So Raleigh, if you could actually maybe focus more on that tested
for
amoeba.
sure. Let me zip it up. And apologies for more of all this scratches on my lens. I need
You
to get
actually look
this.
really good, Riley.
It
It's
looks
coming through
great. Can't
quite
see it.
clear.
Yeah, it's kind of comes
Okay.
fine.
It's a good sales pitch for Omax, honestly. This looks really good.
Yeah, yeah, yeah.
So yeah,
Yeah,
now I'm kind of on that amoeba,
Yep.
focusing a little bit.
hard to say if a vacuole, that's a very large vacuole if that's the case. It does look like more like what Keisha and Casey were talking about, like it's in the process of starting to insist.
It could be a
Um,
vacuole. We have to always remember we're not like, we can't say for sure, right?
Hmm.
right.
Like, it could be that it ate a spore. Sometimes, though, it's like almost certain that they've eaten a spore because you'll see the vacuole and you'll see fungal spores inside of the test that look just like the fungal spores on the outside, but in a more dense number. So you can see that it's collecting them, putting them in its test.
Yeah.
Also, I would say like against spore in this scenario is see how clear it is. You can actually see the aggregation
Hmm.
either whatever that is below it, that little piece of mineral or whatever it is. You can actually see through it.
Yeah,
Uh,
that little guy.
which most spores are actually going to be not so transparent.
Hmm.
Except for some
Huh.
mycorrhizal spores are
I'm trying to
super.
think if there's anything through this aggregate
The
here.
thing to
Oh,
highlight is
bubble.
there's not a lot of I mean, there's so many times when we go through and we're looking at these samples where we're making an educated guess at what we're seeing. Um, and, and there's been many, many times where something that I thought I understood, as I started doing this more and more and more, I started to realize, ooh, maybe it's something different because I found another example, or I read some kind of, white paper or scientific study. And so part of this is also just learning more and putting more hours and time with your eyeballs and starting to see some of these things.
Yeah.
Mhm.
I mean that's an awesome thing about kind of what we want to do with the microscope course. It's like able to look at a sample together and be able to identify
Right.
things together. Because I might see something like I had no idea what that is, and if I don't have some identification book, if I'm not clear, like it's awesome having a bunch of other people who are have a lot of microscopy experience to be like, oh, that's a testate amoeba, and that's looking dry, and like Dan said, is this an actinobacteria right here?
Most likely.
You
Yeah.
know, to use another example of this, I remember many, many meetings where we were starting to put together a lot around the microscopy course, the Sofa Web School, and so forth, where we would sit down with maybe 8 or 9 or 10 of us looking at samples together, whether it be Elaine and other people that are, really hardcore into the microscopy aspect. And the amount of disagreement that would come around when we look at a sample, people are like, "Oh, I think it's this. No, I think it's that. I think it's this." So and this speaks to some of the challenges that we have in the microbial world, particularly, is there's so much variability, so much variance, that it can be easy to misinterpret things. So also don't get yourself so wrapped around the axles, like it's either this or it has to be that, right? It's okay to say, I don't know. I
Mm.
think it's this, and that's what you're going to kind of land on in that space.
I believe a lot of times those are the words that are missed because
Yeah,
when I'm in an environment with people that like PhDs for sure, they
right.
use word I think it could possibly be.
I believe.
I believe. But when I'm on the other side of it, say in Soil Microbe Library on Instagram or Facebook, I mean, speaking with such certainty about what's going on, I
Yeah.
saw recently, it's really cool. It's easy to tell these stories and believe that they're 100% true, but it was a nematode inside of a testate amoeba. And I think the caption was, "Who's eating who? This nematode's in real trouble." And it's like, you can't really tell if a nematode is inside a testate amoeba if it's eating the testate amoeba or if the amoeba is eating it or if it's eating bacteria from inside the test or if it just got lost in there, turned around and is trying to find its way out. We
Right.
never really know exactly what's going on when we're looking. It's all guesswork.
It is.
Informed guesswork.
It is informed, right? Yeah. Hey, Raleigh, I
Oh,
got some fun stuff to share.
yeah. Let's go over to you, Brian.
Testate
Okay.
maybe? Wait, Brian. Let him focus on that.
Right there, yeah.
This guy.
Zoom in on that.
This
Ooh,
one likes to eat fungal spores. I often find fungal spores inside these
The
tests.
big purple testate
Hmm
amoeba.
Yeah.
that's a big boy.
hmm
And this is important as well. Like when Raleigh was going through it first, it was out of focus. You'll find a lot of critters and organisms all throughout that focal plane. So it's really important that when you go into a field of view, and the field of view just means that's what we're looking at right now. This is that whole field that we can see called a field of view. Well, there's a focal plane, which is really from the top of the slide to the bottom of the cover slip, there's a volume of liquid that's there. And what we do is we focus from the top of the slide to the bottom of the cover slip. And going through that focal plane can be really important because sometimes you'll find organisms that you can't see until you get them right in that perfect focus. And that's kind of the nature of some of the transparency that a lot of these organisms have.
All right,
All
Brian,
right.
show me
But yeah,
your
Raleigh,
so exciting.
this looks,
Compost looks
yeah, it
good.
looks good. All
Yeah,
right,
and
I'm gonna
this was like
share.
we were all doing that together as a challenge. like do the backyard compost. And so it's really good seeing that a few months later, even though I thought the pile failed, it's still under the microscope. It looks really
Brian,
good.
is this you?
Yeah.
Will you
This
look
is
at
me.
that
Yeah.
arthropod before you go anywhere else? That's that chunk
Yeah,
I
I'm going
wanted to
to.
check out. It's it's like looks fat.
Oh, the bottom right.
All right.
Yeah.
Yeah, fatty.
So, the arthropod that yeah, Keisha's talking about is right here. It's kind of out of focus for you because I don't have my 40 or my 4x objective completely parfocal with my camera. So it's a little bit out of focus, but let me move over to that arthropod. And a lot of times what happens too, you train your brain. You your brain becomes kind of like a pattern recognition machine where there's certain patterns that as you're scrolling through a slide, your brain will go, oh, that's something unique, that's something different, or you recognize it right away. And so that little round blob with the little tiny leg sticking off there, that was like, oh, there we go. We got arthropod. In fact, when before we started the webinar, we were just testing out the cameras, and the minute this came up, Keisha goes, arthropod.
Oh
Brain
yeah,
saw it
yeah,
right away.
yeah. Well, and you check, right? When you're training your brain, you always go, you can zoom all the way into the 400 times what your eye can see and check and make sure that you're correct. That's
All right,
exactly what
so
I thought it was. Big bottom,
yeah,
little.
exactly. So now I just went from my 4x objective, which is 40 total magnification. Because again, it's remember 4x objective times your eyepiece, which is 10x, which on my camera it's the same thing. I have a 10x there as well. And now I just moved to my 10x objective. So that means I'm at 100 times a magnification. And yeah, kind of cool looking, huh? You can see the legs. Looks like it may be missing a leg? No? Yeah, it looks like it might be missing a leg here. I would assume that this is not a living microarthropod because it hasn't moved since we took the sample. And now I'm going to move to my 40x objective, which means we're at a 100x total magnification.
Well, this is something too I've been seeing a lot lately, just very, very new, is people posting images of microarthropods with just like a clear connection to what species it is. And I don't, but I keep wanting to call the bluff because, we've been working on how to easily identify microarthropods based off of their mouth parts for about 6 years now. I guess has been a conversation, Brian. Do you know who
Yeah,
that
about
is?
that.
What it eats?
Oh, yeah, I don't know.
It's like we have no, it's like it's just something to point out that when it comes to microarthropods, it is not really easy to know exactly what they eat. And in many cases, they can eat a lot of different things. So,
Things.
they could very easily eat plants and insects, or fungi and plants, or fungi, plants, and insects. Um,
Is it is there stuff moving in its belly?
it looks like it's
In its
eating.
It
body?
looks like it, yeah. So maybe it's in some dormancy, or maybe it's just stuff that is died and the inside things are eating
Yeah,
it and it's moving around.
It could be a snack already.
could be a
Hard to
bacteria.
tell. It's definitely not moving. But what else do we see on this particular example here? There's some testate amoebas. There's one right there.
Oh yeah, hiding.
And then can you guys see one? There's one lower. Let me go down.
You see some actino too.
There's another. Oh, and I should have mentioned what this sample is. This sample is from my vermicompost bins. So this is worm bins that I have that have been going on for, I've had them for about a year now.
Nice.
All right, so I got some other things to show you. So a common practice for us is a lot of times when I get a sample, I will use my 4x objective and I will scan the entire cover slip, all the way through. And what I'm really looking for is like the microarthropods. I'm also looking for nematodes.
Big part.
And also just something that might be unique. Sometimes you'll see an area that has a lot of fungal wefts, like it's a dense amount of fungi. So that's an area that I'm going to want to be able to go look into and observe. And this is a little bit different from the qualitative to the quantitative, because in the quantitative, you want to be very randomized in your fields of view, which means I'm not biasing or pre-selecting anything. I'm going to pick so many fields of view and I'm going to do my counts through there. When I'm doing a qualitative assessment, it's much more of, I use the word adventure, but it is that way. where you're going through and you're finding things of interest. And I'm like, oh, that's interesting. Let me go take a look at it. And the 4x objective helps me kind of orientate to what are some of the things I might want to see in this particular slide. And then I'll use my 10x and 40x objectives to really go down and look farther.
What's that wiggling
So
on?
oh, what, it was something wiggling?
Yeah, while you're talking, I felt like it was really moving
Could have
along.
been, there's some really cool ciliates in this one.
Okay, could be.
I find in my worm bins, especially, I find some really neat ciliates. I do have a nematode right here, though. Everybody likes looking at nematodes. All
Nice.
right.
If it's not a
You guys see it?
tardigrade, I'm not interested. No.
Exactly. So let me zoom in here.
Sweet little fungal feeder, is it?
I think it's actually a
Oh, it might be
bacterial.
bacterial feeder.
Yeah. That what I'm getting here is hard for, and this is something too, even though I have a really, really nice camera, and I get really good visualizations from it, the best still for me is I can see more detail when I look through my eyepiece than I can through my camera. Um, and so one of the things that I'm going to focus, try looking in this area right here. This has a terminal bulb, and there is a cuticular valve there. And that's usually a dead giveaway for me that it's a bacterial feeder.
The mouth too, when you
Really,
is
yeah. And the mouth has that little tiny, tiny bit.
Mhm.
Right here's the mouth part. There's a little tiny narrow chamber. But this is a small guy here. I'm heavily zoomed in here. It actually almost has more like an omnivore mouth, to be honest with you.
Yeah, on my side, it's not, I'm not getting like the crispness of the mouth unless you're actually
Yeah.
moving it. Even the valve was like, I can see it right now, but it was hard to see at first.
First, yeah. I've got another one that I saw earlier in here. Um, it was an omnivore.
Big wide open.
See if I can find it again. Oh, we got something interesting. See, this is what happens when you're doing the microscope adventure here. I wanted to find that omnivore nematode, but then I found something else. I think this is maybe what you saw earlier, Keisha.
Rotifer.
Rotifer.
Yeah.
Oh, yep. There's this hold fast.
Yeah. So you can see the hold fast is right there, right?
I think I see some
And
flagellates.
then wait until these things are so cool. They're one of my favorite organisms to watch. Is once they decide to put out their rotors, then you see that massive amount of vortexing happening.
Come down just a little bit, Brian. Your view is slightly different than ours. Right
Yeah,
there.
I think I gotta unfocus a little bit. And then they have a little pump on the inside of them that does a really good job. Let me zoom back out a little bit. There we go. Did you guys see the flagellates moving around here as well?
Yeah, I kept seeing him
Yeah.
bumbling
Yeah.
in. Well, aren't
Well, we'll come back
rotifers
to Mr.
aerobic
Rotifer. See,
organisms, Brian?
say what?
Rotifers anaerobic?
Anaerobic.
Yeah, you'll tend to find them more, I think, in the anaerobic soils, but I've had plenty of samples that were from pretty aerobic situations and still found rotifers in there, kind of like ciliates in that sense.
I,
I
I
like
was
a concern if I see a lot of rotifers. You know, if I see one rotifer in a slide, not a bad. There, oh, there we go. See the rotors are going. You guys see
Nice.
it?
That's cool.
You can see the vortexing happening around it too. And then
Mhm.
look at the inside of right here. My understanding of that is that it's like a crushing, like it's pulling material in, and then it's like crushing the bacteria as it goes through.
Yep, digesting it.
Yeah, such a cool
Yeah,
organism.
yeah.
Right.
Rotifers, and you can see all the look at all the vortexing. You can see the bacteria just kind of moving around.
Rotifers are unsung heroes when you're talking about micro livestock. Um,
Yeah, I agree.
these municipalities actually breed rotifers to take care of human waste. They're very,
Right.
very good at eating an abundance of bacteria and cleaning up really nasty situations. Um, they're hardy critters.
They are very hardy critters.
You know,
Yeah.
when you're like you think about like a copepod, like you would never see a copepod in a toxic environment, but these guys,
Right. they can handle it.
they can
It's
handle it.
fine.
Yeah, yeah.
Let's see if I can find that nematode I was talking about earlier. I thought it was down in this section of the slide. It was a big omnivore. You can see my slide's starting to dry out a little bit here.
Dry Island.
You know what I'm
And
realizing
this is
on timing?
go,
We're gonna run out of time early on this one. If you guys like have generalized questions about microscopes or anything as we go,
yeah,
those
please.
in so we can answer them.
Brian's dilution ratio is 1 to 10, right?
Is it Brian on this one?
This one actually, I looked at it. It's a 1 to 8 that I made on this one. I'm not too concerned when I'm doing kind of the adventures of getting to a specific um, dilution rate. Although I do have another two samples that I did make specifically at a 1 to 10 dilution because I want to compare the two. So even in a qualitative sense, sometimes the consistency of your technique is really important. If I want to compare like the two soils I have is one from my garden bed and one from an unimproved area of my land, I wanted to have much more of an apples to apples kind of comparison. So I made them the same dilution ratio so I can see the differences between those two. But there's other ones like my vermicompost. I really wasn't worried about comparing anything. It was just more to look. I just wanted to find a good dilution rate that allowed me to see a lot of organisms easily. And I chose 1 to 8, and it worked really well for me in that sense. I got another nematode here. And you can see here, this is hard to see. The mouth part is definitely hidden. All right, and also a question for you guys out there in the webinar. Do you see that curly Q thing over here? All right, we're looking for the first response. What is that? It was one of those things that tripped me up in the beginning.
I get
Stacy got it.
I get
Cellulose! Woohoo!
yeah.
Good job. Good
Oh yeah,
job. Yep,
people,
it's
everybody
cellulose.
got cellulose.
Hey, so there's a question here, like, can you please explain how you do your qualitative analysis and how you document them? So I'll start, because I have
Sure.
a lot of different formats that I use depending on where I'm at and like what job I'm working on at the time. Um, One of the qualitative analysis that I really like that is like a super, super quick check. It's something that I do when I'm at events. So often we'll be at whatever conference, and the people at the conference will send out an alert before the attendees come to say they can bring their sample in, and Catalyst Bioamendments is going to look at it for free. And what we do is we put up a big screen TV, and then we have them stand in a line, and we have like sample after sample we go through. And I try to give each person something that they can actually take home and have some sort of documentation of. And I should have this at this webinar right now, but I don't, because it's just a printed out sheet that I use in real life. I don't think this is the first time I've ever teaching this, but basically it's just a piece of paper that names all the different functional groups and then has a place for notes. And I go through, it's already pre-printed. So there's a little blurb in each section about what, the light idea of each functional group is, like what purpose they serve, fungi, creating structure in the soil, bacteria breaking down, And what I do is I go through each different group of organisms and I just tell them what's present and what I see. And then at the bottom, I'll go through and give an idea of, how they could go about increasing their microbial reality. Now, whenever I'm talking about clients, we have something we call a quick check that again it's almost like a spreadsheet with just larger cells in it. And I go through and talk a little bit more in depth about everything I'm seeing. I oftentimes add photo or video if they're interested, and it goes into a file. But the reality of working with clients, I mean, in the work that I do, everybody that's out doing their consulting or has their lab work, it's different. We each have our own reality that we've created because this isn't a normal job, is it? There's no one telling you how to do this work or where to apply for a job like this. But a lot of my clients, when the first time I look at their sample, there's nothing. Having to dilute it 1 to 10,000 to count bacteria, 1 to 2,000 to count bacteria. Um, so my notes are pretty sparse. And oftentimes, what's going on is there'll be someone on site with a microscope that is kind of my counterpart. They're their boots on the ground. And they'll start looking under the microscope, and I'll ask questions like, hey, are we seeing any predators? Nematodes or testate amoeba, naked amoeba, flagellates? And often the answer is no. The very first 2 or 3 applications, we might not see any movement at all. The day that the person calls and says, hey, I found a bacterial feeding nematode and 4 testate amoeba in the sample, that's a lot of data. It's huge actually to understand that the microbes we're taking from the compost have finally made their way into the soil and they're starting to reproduce. So yeah. Brian, do you want to talk about how you present qualitative, or do you ever present qualitative? You don't really, do you?
I do present qualitative, and it's very similar in that sense, which is usually you're looking for key indicator organisms. And in fact, I've got one of my clients, a pistachio farmer, where they're learning how to use the microscope. And one of the things is just for them to be able to check. A lot of times I tell clients that at the end of the day, you're becoming a microbe farmer when you're doing this kind of work. And if you don't have any ability to observe or see or understand the microbial communities that you're farming, it becomes sometimes really hard to manage them. So having a microscope just so that you can go out there and look and see is important. And for, let's say, that particular client, one of the things that really stands out for me is we're going through the process, and all of a sudden it was like, I'm not seeing the organisms that I was seeing before. Like they had already internalized and thought that. So when we looked out there, we started seeing a lot of the protozoa were insisting. So it was giving us an indication that something was happening in that sense. And so their qualitative analysis triggered a discussion that then led us to do more detective work to find out what was really going on in that particular scenario. And really it became because they were spending enough time observing that they were able to intuitively understand that something has changed, something has become different in the sense.
Okay.
Right.
Yeah, so we got a lot of good questions coming in now. Um,
Oh,
we could
whoa, there's
share,
tons
you know
in
what I
there.
might do? Isabel, I don't know if you follow us on Instagram. That'd be a really easy place for me to post some of my different sheets when I get home, and I wouldn't mind doing it. You can find us at Catalyst Bioamendments on Instagram or at compost.academy, I think. And we're doing promotions right now, so why not? I'll share some of our documentation. I actually don't have it on this computer and I'm traveling, so I'm gonna have to take a picture of it and share, but I'll get at that. Um, Ollie. Also, somebody asked here, at higher magnifications, I see a lot of jittering, vibrating dots. Are these bacteria, Brownian motion, something totally different? Apologies if we mentioned it earlier. We didn't mention it earlier, and I think you're right. It could be all of those things. Like Brownian motion is kind of the movement of, the vibrations of the earth and the things around you. But there's also things called flagellated bacteria that move a lot like flagellates. And then there's also, lactobacillus. Most of the bacillus species have their own little wiggle and movement that they do. So, yeah, all.
Oh,
All the above. And sometimes in the samples too, like in this is vermicompost. So really small clay particles are going to be not nearly as prevalent. I mean, there's organic matter that's getting broken down. It gets really small. It gets that kind of browning motion. But when I'm dealing with a heavy clay soil, that's where it becomes really difficult to understand because the vast majority of those small vibrating dots could be clay platelets and not actually bacteria. And this is where you would have to make a much higher dilution to really get down to where you're only seeing, maybe 10 of those particles on your field of view. So you can individually look at them and say, does it look like a cocci? Does it look like a bacillus? Or does it look jagged and irregular? It's probably some kind of either organic matter or clay platelet.
Brian, do you have a blue tarp in your yard that's made that microplastic in the sample? Somebody busted already.
Yeah, no doubt.
I
That
see.
is microplastic. I don't have a blue tarp, but this is coming from a lot of food waste
Aha.
into my worm bins, and so it could easily come in from any kind of source.
Sticker.
Like a
Okay.
sticker on some material, or who knows.
We're getting a lot of good questions, but I feel like Raleigh, micro sapphire. I
Mhm.
wonder what they're worth. Raleigh, do you want to do the promotion really quick just so these good folks know about the microscope program that's
Yeah,
coming
of
up?
course. Let me
Let me stop sharing here,
share the screen real quick. All right.
Microscope.
Yeah,
and I will find another sample for us to take a look at.
Awesome. Well, folks, this is what the reason we're putting this on is we were really excited to put on a program that's going to take people on a journey for 6 weeks of training. So it's basically like you get a pretty much the equivalent of like a virtual apprenticeship on microscopy. And we have this program coming up December 11th that we've lovingly called the microscopy course. So I think one reason we really wanted to put this together is put people on the fast track to learn how to actually analyze their biology, how to see the quality in their compost and in their soils. And even though, a lot of us start out, we have our own self-pay training, it helps so much to be guided along by folks who have do this as a profession to help you understand what you're seeing, what to overcome some of the challenges you have with microscopy, so that you don't feel like you're just treading water by yourself and you actually have guidance on learning how to do, proper microscopy and some good qualitative analysis. And kind of the features of what we're doing is, of course, we have this live group coaching sessions. That's 6 weeks of training with Brian, Keisha, Casey, and I, where we're gonna walk you guys through week by week. You're welcome to bring your microscope along, share samples just like we're doing. We can talk about what you're seeing, patterns you're seeing, and of course, each week is kind of a different focus as we guide people kind of along their microscopy journey. And of course, another big part of it is we did film. We have self-paced lessons coming up to show people like how to use a microscope, orientation to the different parts, kind of some examples of soil and compost that we're analyzing. And another fun thing is we have challenges. So like we'll have challenges that'll show, you'll have action challenges to do microscopy with yourself and share your results. So it's fun thing that you're not just showing up and there's nothing to do. It's like, no, you will be challenged to actually do something and share your results. So you're not just absorbing information and theory, but you're actually doing something with it. Another great part that, if you're part of our other program is we have a big online community. So people from all across the world are sharing their results, sharing the organisms they're seeing, doing the challenges with you. And we all share resources and guides, things like the right microscopes and equipment and all the things that you might need to do microscopy in a really good way. So a little example about the schedule. This is tentative. So week one, it's December 11th. It's kind of foundations where we start, why we use microscopes, equipment overview, and setup and support. Week two, techniques and troubleshooting, practicing slides, samples, addressing common issues, building confidence. Week three is when we really uh, start really identifying, jumping into organisms. Week four, bacteria and protozoa, spotting and differentiating the microbial groups, connecting their connections to the soil food web and soil health. Week five, we start going into the really bigger organisms, nematodes, arthropods, and more. Identifying the higher trophic level organisms, understanding their role in the ecosystems. In week six, January 29th, soil comparisons and student practice, viewing samples from totally different environments, practicing full observation, sharing findings, and reviewing your guys' needs, resources, and next steps. So if you want to join that, it's available right now, and it's coming up. We're going to start in December 11th. And right now it's $750 if you're not a Compost Academy member. If you are a Compost Academy member, it's only $600, so sweet deal. And I'll drop the link in there. And of course, if you just want to sidestep all this, we have a program coming up, the whole Microscope Academy retreat. It's happening in Costa Rica, February 1st through 7th in Rancho Margo. Last year, we had an incredible experience with 30 students. We spent 2.5 days on microscopy. So it's a really good time to get together, learn how to make incredible thermophilic compost, make extracts and teas, and actually get a lot of hands-on experience with microscopy, and do it in a really epic vacation venue. So if you want all of this and more, you got to come to the Compost Academy retreat. We need to get 10 more people to sign up by next week, and it's a go. So we're going to be emailing out people about that. But
The Costa Rica one's super fun because the microscope adventure is insane.
Insane.
yeah, yeah, the stuff you
If you
see there.
first of all, like you can see with your like there's just so many animals, so many mammals, lizards. Like for me, that was just the best. The baby sloth, all that. But we started
Right.
thinking, we're like, man, that sloth is like covered in biology. You can see it. The sloth is literally dripping with
Got lichen.
ecosystem. It's got lichen growing on it. We're like looking at all these animals and like, man, I bet they spread the microbes everywhere. And we actually like we were sampling leaves, sticks, like we were sampling the craziest stuff and finding nematodes and arthropods and stuff that we didn't know. Lots of stuff we didn't know. Really, 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 really fun. So,
Well, and the microplastics, that shocked me. We're out in the middle of the tropical rainforest, and man, we were finding microplastics all over the place. It was kind of
Are
crazy.
tons of
yeah,
time.
lots of tarp out there in the
Yeah.
jungle. But then also the microscope class, just to add in some like from our point of view, this is going to be the first iteration. So to be frank, it'll be the cheapest. That's how it's been so far. As we go, we continuously put more and more and more information, and we build these classes out till they get bigger and more. But once you're in, you're in. So as we continue to change it, which we will, you'll be there along for the whole ride. So we have a lot of folks in our other circle, which is more about composting and less about microbes, although we do have a little bit of a microbe thing going on there, little microbe library. Um, some folks are on their third, they're going to be going on their third iteration of the class and just, starting with a goal and strengthening their information as they go. So this isn't just a workshop that you get into and have to accomplish within, 6 weeks of time. You're on the team. You'll be there for the duration until either we stop working or the internet explodes. So
Right, right.
it's a
Yeah.
forever class. So you don't have to necessarily participate now. We'll be at it for years to come.
And the
And
good thing is it's not just AI. So
Yeah.
Yeah,
not just AI.
I
exactly.
haven't started using AI for our content yet.
Yeah.
Okay. Oh, Susan popped her hand up. I'm gonna allow. Hey, Susanna.
Do you want to speak?
Oh, we're gonna
Yep.
wait for her to unmute that microphone. Susanna, if you can unmute your microphone, you can ask your question live. I'm going to.
I think it's for anybody too. If you want to ask a
Yeah,
question
yeah,
live,
totally. If you
by all
have a
means, raise
live
your hand.
A
question,
lot of good questions as
raise your hand.
on the side
We love
live.
live questions.
They
Yeah.
do.
Well, it looks like she had trouble with her microphone. But if anybody else wants to ask a question, just raise your hand in the, in Zoom and we can get to you. But in the meantime, we can go to some other questions. Keisha, do you
All
see
right, and I'm
anything?
sharing some stuff back out as
Oh
well.
yeah, go for it.
Sweet.
So one of the things I did want to show you guys, and this is a little bit about the qualitative analysis and some of the questions that came before, which was, how could you use qualitative analysis to do some comparisons? So here in my yard, I have two soil samples that I wanted to compare against each other. One is from an area of my yard that I have not done anything with. In fact, it's a highly disturbed soil. We just put a greenhouse in this area. And I wanted to look at that versus one of my garden beds that I've actually been spending time trying to increase the microbial community and build structure in the soil and that kind of thing. And this is where I can really make some very valuable assumptions about how things are going for me between looking at those two samples. So, in this sense, because I'm doing a comparison, consistency is key for me, which means that when I made my dilution, I made the same dilution ratio between the highly disturbed soil and my garden bed. So I can really make kind of an apples to apples comparison. And when I mix them for a set period of time, usually 30 seconds for me to mix a sample. And then I waited 10 seconds for the sample to settle before I actually drew it. So I wanted to make sure that my technique was consistent between these two samples. And this is similar to what we do when we do a quantitative. We want to make sure that we are consistent in the technique. When I'm doing my adventure stuff, I'm not nearly as beholden to a technique. I can let myself be much freer in that sense. But because here I want to make a comparison, I chose to do that. And so this is the sample that is from the highly disturbed soil. And a couple of things that really kind of point out to us is there's not a lot of aggregation here. In fact, you see a lot of mineral material through here, although it's not devoid of life. In fact, right here, this is a fragment of a fungal hyphae, right? So there is fungi growing, and I got another little fragment over here as well, and you can see that right here.
See how everything's mostly clear, the background, like most of the
Right.
mineral, it's just like you can see right through everything.
It's
That's
shiny and
Yeah, you got
sharp.
dark colors. It's speckled. There's teeny little
Yeah,
bits speckled throughout there, but in the last sample, everything was mostly amber and brown, and it was like the inverse, right? You saw a lot more of the browns than you did the clear stuff, and this one you're seeing a lot more clear.
exactly. And so, and this is common for when I look at highly disturbed soils, like for my clients, who are especially like doing like, let's say, large scale agriculture. In fact, this looks better than a lot of their fields do, which is here I'm starting to see some aggregation that's occurring, and I see some fungal hyphae. I'll sometimes look at soils that will be really devoid of almost any microbial life besides maybe some bacteria. And in here, this soil, it's just kind of wild. It's a lot of early sessional weeds grow here. The soil does not have a lot of structure to it, and I can already make some of those kind of very impressions about that. So let me switch to my garden bed.
Yeah, while you're switching, there's a question here.
Uh-huh.
So actually the qualitative thing is more for clients. No, the qualitative thing is for people that don't have a lot of money, or don't want to spend a lot of money on testing. Because, there's so many different tools in the tool belt of how we can look at microbial reality in the soil. I mean, we have our microscopes, and even within the microscope world, how many different microscopes are there? There are electron scanning microscopes, there's compound microscopes. Within the compound world, there's a lot of different tools we could use. Um, we can look at DNA analysis. There's so many different tests that we can use. But honestly, most farmers, most of my clients are not coming to the table with, say, $5,000, $10,000 for testing. If anything, they just want to have a little peep at what's going on and then check in as they are doing their work to see if they're actually affecting change in their ecosystem.
Hmm.
So it's a cheaper way to see what's going on. If I'm
Yeah.
counting every single time, it's basically $125. My price per sample doesn't go down until you buy 10 samples at once. So, yeah. And for me, like, I could make that price less, but I don't like to do it, and I don't find it to be necessary for the work. So I keep the price high so that I'm not doing a bunch of work that I don't like.
For sure.
I'm just being frank here. This is like, this is my job every
Yeah.
single day of my life. Like, this is how we operate. So, oftentimes, like I do my compost, we use a quantitative,
Yeah.
because that's what everyone wants to see. We've built Catalyst bioamendments. Our client base, they almost all use microscopes. Almost all of them are, Soil Foodweb students. So they're very tuned into those numbers and they want to see those numbers. And our numbers are good. So, it's great to share it. But in reality, how often am I using SMAP? Not very often. It's usually qualitative. So,
Right. Well, and you spoke to the cost thing too. From a cost from my client standpoint as well, is I can teach them for a relatively cheap, versus a lot of testing, how to do qualitative assessments on their own. And they can do a lot of the qualitative work themselves. And in fact, that's a really important thing for them to, again, being that microbe farmer. I always use it to the analogy of if you were a cattle farmer and you never saw your herd, you never observed it, you never looked at it, it'd be very hard for you to determine how good your herd of cattle is actually doing. It's similar in that sense when we talk about the microbes that I want my farmers to be able to observe and see and then become very familiar. And qualitative is accessible for them. Teaching somebody how to do quantitative analysis is a much different ballgame. And it's a much different commitment when it comes from their standpoint to do something like that. So
Yeah. There's a
it
good
just works
question
well.
here from Kenneth wondering if we could post, yeah, a few people have asked this. Can we post the qualitative assessment report, what it would be like? Is interested in joining, but wondering what the assessment would be like. Yeah, I mean, I can post the assessments that I use at Catalyst for sure. I could share
Sure.
those. I mean, to be really honest, like we are still, we've probably got another week before our final template is developed. But I could maybe it would be smart for me to just spend a little bit of time showing off what we've done so far and maybe uploading that onto Instagram or maybe I don't know if you guys are all in our email. You are because you got a link to this. So you must have signed up for our email group. Raleigh, maybe we could put something together and send an email around in the next few days. So at least folks can get an idea of what the general assessment will be. I know we're going to play with it a little bit because that's how it goes when we're building out these templates. We're always adjusting them to make them more student friendly. So for like a good example is we have a nice template that we have for building our compost recipes in our other circle, not the one that we're talking about here. It's the compost focused one. And that template has been changing constantly over the last two sessions because students keep coming up with things that they would like to have different or coming up with like issues in the format. And so we're constantly sharing, but we'll try to get some sort of idea of, a basic idea of what it's going to look like. And someone else asked, like, does the course allow me to give official reports and to have an official laboratory? The word official, that's where I would say like I am a soil food web lab and I have a soil food web lab certificate. And I cannot be an official lab until I go to, I believe, I'm not sure if it's a state office or a federal office that you have to actually apply with to be an official lab to be able to take samples and, terminate the life in them. But you can be a lab anytime you want. You don't need certification to be a microscope lab. It's a made-up job at this point. It's not really, the idea is like you look at microscopes, you can provide reports. If you can get people to actually work with you, it's an official lab. And I say that because that's really about as valid as any of our microscope labs are. We're not official in any way, shape, or form, unless I'm missing something. Brian, do you know more about the officialness of your
No,
lab?
yeah, I think you're spot on. I mean, the intent of our class is not to for you to walk away and say, "Hey, I'm now a Compost Academy certified lab tech" or something like that. It really truly is about how can we teach you the tools and techniques so that you can become much better at using a microscope, and then be able to do the kind of qualitative analysis to be able to say, "Here's my impression as far as what I'm seeing in that sample." Um, so yeah, just to be fully transparent, yeah, we're not offering any kind of certification or anything else like that. And I think there's I mean, obviously there's some challenges like Keisha was mentioning. Are you becoming state certified? Do you have to look at the regulations if you're going to say that you are going to become a certified lab? There's a lot more complexity to that. And there's potentially a need for that, but that's not the need that we're trying to fulfill in this sense. We're trying to just teach people how to use a microscope and how to be able to assess what they're seeing, and then provide that feedback if they need to somebody.
Yeah, I would say that we have a strong interest in developing this career path more. I mean, all of
Mhm.
us know what it's like to start work. I mean, Brian transitioned from tech, which is an incredible job, really good money, into soil food web work. Casey and I made our own transition. So did Raleigh. We know what it's like to go out and like try to make this your job. And we have if anything, that's what Compost Academy is for. But I hate to make any sort of promises and say, come to our school, and when you come out the other side, you're going to make $80,000. Because that might be a lie. What I could say is like, if there is a strong urge from the students to have a certification, for us to say, Compost Academy validates that this person knows what they're doing, we will get there because we are very interested in what you need. Right now, there's not an overwhelming desire. Brian and I are doing this microscope class because we like it and we want to and we think it's fun. And we're actually trying to drum up enough interest to even run this program. Right now, most of our interest is in composting. Everyone wants to compost. We want to do a microscope class, but this could turn into something where we say, like, yeah, this is a lab. officially through Compost Academy, it still won't make you an official lab in the real world. We would have to do more research and help you guys figure out how to do that, you know? You need an autoclave, you need to know this. And, that's the kind of research that we could do. We could put together that kind of program if that's, the overwhelming desire of the team. Because at Compost Academy, that's really what we're about is our students and making sure that their goals are fulfilled. So this really is just an adventure on this go round. We're probably going to do way more in-depth stuff. I have big plans to partner up with a lot of people in the future, but honestly, that might end up going into podcasts. So we've got so much going on right now. Yeah, I guess
For
one
sure.
bigger.
Yeah, too. I think a really big component versus just like, a lot of these programs, you do a self-guided program and you might come out with a certificate, but still, it's about putting the hours in under the microscope. And I know for me, it was like I did the self-paced, like Soil Foodweb course, but then I was like, God, I need to learn more. So I actually went to Todd Harrington's lab and just lived nearby for a month and traded. I helped him make his website in exchange for getting to work at his lab, and it was this really great experience of getting to like test out different microscopes and look at different samples. And then when I got back, I was actually like paid for consultations with Joe to help train me on a microscope. So
Yeah.
in the meantime, this is like, wow, to do this with a group for 6 weeks, this is the program that I wish I had to build that kind of experience.
Yeah, and I think we're fulfilling that particular need, which is, there's plenty of times I'm advising my clients, I would really like you to purchase a microscope and learn how to use a microscope to get in that whole microbe farmer kind of aspect of it. And just that level of education and knowledge and training is what's really needed for me for a lot of my clients. And so we think that there's a lot of other people out there that just need that kind of level of skill. And that's what we're trying to fulfill the need of.
And Natalie's saying like, and this is probably true. She says you need your university degrees to actually be able to open an official lab in Central Europe, and it's that's probably true. And what's really funny about this is like I don't have a traditional degree at all, like not even one little teeny one. I tried college and was like university and was like, whoa, they're telling me what to do. No, I want to study microbes. And I now get hired by universities to develop their microscope
Right.
program. So it's a funny thing. It's like, the girl that doesn't have a degree is, at the university teaching
Right.
the microscope department, like what kind of microscope they need, all about
Sure.
identification, helping them develop a way to make it fun for the students so they want to do it. But it is like, there's already a world set up around this, and it's like, it is very like traditional university based. So we'll see how that all changes through the years. I think there's so many cool programs going on online that actually give you in-depth
Right.
information on what you want to learn.
Right.
But yeah, now I want to look into what you have to do to be a real lab. I don't know why I never.
Exactly.
I'm feeling
So
ignorant.
I wanted to jump back into this, that comparison I was talking about earlier. So this is my garden bed. And a couple things to observe. I mean, right now you're seeing a lot of aggregation. So you can see as I'm kind of moving my focal plane through those
Sorry,
aggregates.
Brian, I
But,
accidentally took over your screen share. I'm going to stop sharing and let you go
oh,
back.
sorry.
There you go.
Um,
Probably had a lot more testated amoeba than you.
oh, did he? Is some more fun stuff? Well, hey, Raleigh, I'll
More
pass it back to
fun.
you just 1 second.
No,
I'll
no,
just
please.
finish this.
Yeah.
So this is part of that qualitative analysis that I look at, which is so this sample, I'm seeing a lot more aggregation. So you can see the aggregates, right, inside the soil. And again, this is my garden bed. Same dilution as the disturbed soil, same technique and processes, but you can see some differences. But also notice that there's a lot more kind of open space, not as much mineral, just kind of evenly spread out. Well, that's because a lot of these minerals are the sands, the silts, the clays, and the organic matter is getting pulled together to make these aggregates. And so already when I look at the sample, I'm making some impressions and saying, all right, this is definitely different. So I've built some structure because I've got aggregation, and I'm also seeing life. Like right here, here is an amoeba, right? At least I think it's an amoeba. It might be a really big flagellate, but I don't see any flagellum. Oh, I do see a flagellum. It is a flagellate, a very large one.
Oh no.
Look at that. But yeah, so this is, this gives me some good impression, and also you can see some of that better fungi Starting to see more fungal hyphae in here. I think I have potentially an Arcella right there.
Oh yeah. Arcella can have some different shape to it. Could be
Oh, and
Arcella.
you see the
No,
empty
you're
test?
right. There's something weird, huh?
Yeah.
Oh, I
And
do
there's
see.
the empty test right here. So, but overall, just in this particular example, because I was able to compare these two samples together, I'm already making some good impressions. I'm seeing some constituent organisms or the key indicator organisms. I saw a flagellate, there's amoeba, I'm seeing fungi in here. I'm seeing aggregation that's occurring. And so for me, that is telling me a lot of information about how that soil is progressing. And they came from the same source. I mean, these are two soils. My garden bed was just the native soil here that I've improved over time by adding compost and mulches and adding plants in the equation, versus the highly disturbed soil, which was just a patch of early successional weeds, that was an area that I haven't done anything with yet. So um, that is a lot of powerful information without even doing some kind of quantitative analysis. It's telling me a lot of things about the soil.
Are you at 400?
I'm at 400 right now. Yeah.
He's at he's more in.
400 total max.
So let me
Um,
zoom in that flagellate. Let me see if I
you
can
guys,
find it
I
here.
hate to do this because we always just stay on as long as we possibly can because it's so much fun and I am having a great time. But we are promised to be out of here at 12:15. So Casey and I've got to
Oh,
go.
okay.
Is it okay if we leave and you guys stay on?
Yeah, for sure. We can stay on a little
I
bit
feel like
longer.
I don't want everyone to miss out on all this testing to me, but that was such a great
I
sample.
know, Ollie has a
And
great.
Brian's showing such cool stuff. Will it mess anything up if we just exit out and go?
Well, how
Ah,
about
I don't
there's
think
one
so.
last question from Liz. Well, actually, no, you guys go. I bet Brian can take this one.
Yeah. All right, you guys. I
Sure.
hope we see you at the microscope class, and if not,
If not, we'll
we'll
see you in Costa Rica.
Costa Rica
Yeah,
Yeah,
exactly.
or at the
get post the
promotions in the
emails.
future. Or yeah. Thanks for joining
Thanks,
us today, you
everybody.
guys. We'll see
Peace
you.
out.
Ciao.
All
Bye.
right. Well, Keisha
So
and
we'll
Casey
hang on
are
for
popping
about another
off.
15 minutes or so,
Yeah, yeah.
and take additional questions. And Raleigh, you can share out what you want to share out. But there's a question from Liz in there.
Yeah. It's like, how about portable setups to more easily do these with farmer clients at location or the farmer's market? Any thoughts on good microscopes and supplies for this approach? So portable setups for demonstrations.
For sure. So a couple things about this. I travel a lot with my microscopes. For my really good microscope. So let me just show you right there. That is my LW scientific microscope. It was a fairly expensive scope. It's got, it would be hard to replace a lot of the parts, economically. So I want to protect it when I travel. So let me just move back a little bit here. You guys see this? Raleigh, is that coming across all right?
Oh, yeah.
So this is a hard case that's got foam cutouts that are very specific to this microscope. So the microscope fits very tightly in there. Plus all the other materials that I need to travel with go in that hard case. And that goes with me especially when I go on airplane flights, because they're going to get thrown around. I want something that's going to really heavily, heavily protect it. Now, give me 1 second here. I also still have my OMAX scope. And in fact, for probably the first 5, 6 years, I used just a low end OMAX to do all of my work, even when I was doing it professionally. And in this case, I use a tote. I'll show you that.
Oh, funny.
So you can see it's just a tote
Yeah.
that's got a microscope, and I stuff it with towels or rags to help with vibration as it moves along. And that OMAX scope, I usually take out to me when I go out to clients' locations, especially if I'm going to be doing samples in dirty, dusty environments. There's many times when I'll go out to a farm and they'll be doing, let's say, an almond farmer, and they're out there doing whatever work that's creating a lot of dust. I don't want to bring my LW out there. I'm going to bring my OMAX because it's good enough for what I'm trying to do. So, yeah, I find that just a plastic tote with your Omax and just put a lot of towels around it to kind of hold it in place works just fine. If you're going to travel, again, like I said, like an airline or airplane, where you're going to be having, a lot of movement with the microscope, you invest in a hard case. It's well worth it, especially if you spent quite a bit of money on an OMAX scope. I mean, sorry, like an LW scope.
Yeah, without a doubt.
And
I've
I
got the
do.
one with foam padding. It's like having that foam padding helps so much.
It does
Yeah.
tremendously. And I've done a number of events where I'll show up. And really, for me, the kit that I usually use when I go is my microscope with a folding table. The biggest thing before you go do something like that is the table and the chair. Make sure they're set the right height so that you can easily see use the eyepiece kind of thing. And for me, if I'm going to be going to like a farmer's market, you need some way to be able to visualize it. So I'll bring a TV along as well, and I'll hook my camera up to the TV so that people can see it in a much larger scale. And it works really, really well. I've had great success at doing small events and things like that by just having a TV and a microscope and a table and a chair, and it works.
Yeah, getting that video attachment is super important. I know some people can, they can like hook your camera up to the microscope, and I know like Keisha and I did that to film, but I just really like the setup where yeah, you just stick your video attachment in, it's
Right.
USB, and they're just what we're doing now. We're just using like tube view.
Exactly. Like right now, I have it hooked to my laptop, but you can hook it to a monitor, you can hook it to a TV. There's usually a lot of options when you're looking
Yeah.
into that. And it's a great question because there's a lot of people who want to be able to show the life in the soil, the life in the compost, and things like that. And for fairly easy setup, it's very powerful. The visualization is really, really important. All right, Raleigh, what are we looking at?
Hold on, I was trying to find. I zoomed. There was one. There was like the land of amoebas here. I'm trying to find it. There was like just a gazillion in one view. But here's this one interesting thing. I'm like, what is this little brown corn cob here? Is this like a fungus?
That you zoom into is probably some amount of organic matter, I would assume is what
Oh,
it
okay.
is.
Yeah.
So you can see the individual cells that are there and it's breaking down. That's what I would assume it would
Okay, got
be.
it.
Yeah.
But it's cool seeing in this view. It's like, oh wow, amoeba, amoeba, um,
Yeah.
amoeba.
And let me say one other thing too about qualitative assessments. It's really important here. So let's say, Raleigh's got this compost and he wants to be able to make extracts or compost teas or use the compost in a potting mix and those types of things. Well, with really high confidence, after Raleigh looks at this compost sample, he can basically make the impression or interpretation that that compost is chock full of really good microbial life, the things that I want to be in my extracts and my teas or in my potting mix. So if I use this product, I should be able to inoculate, right? I should be able to move those microbes into that growing session. On the flip side is, let's say that Raleigh went out and he purchased a bag of compost, and you want to look at it. And if you looked at that compost on a microscope, you can really quickly determine, oh, wow, it's missing fungi, it's missing protozoa, it's missing nematodes. So if I try to use it as an extract or I try to use it into making a compost tea or as part of my potting mix, I'm probably not going to get that much of a microbial boost, or, I'm not going to do what I was setting out to do, which was to create that microbial community. So the microscope is essential in that kind of aspect to be able to make those determinations. And then on the flip side is once you've done the extracts or the teas, or you use the compost as like I would say a potting soil potting mix, and you then look at the soil, but you're not seeing anything as far as microbial communities establishing. Now you have saying, hey, I know I use known good product or inoculum, but I'm not seeing any of those microbes starting to establish in my soil profile. Now you got to go determine, well, what's happened? And this is where you put your detective hat on. Why am I not getting a microbial community? And now you start asking a lot of questions. Is it my water source potentially is causing a problem? Am I applying some other product that's potentially killing the microbes? Is it contamination in that soil already? And so the microscope is, again, is a very good diagnostic tool in that sense. All right, Raleigh, you've seen some good stuff here, some testing amoebas. I think I see
Yeah.
a flagellate maybe moving around.
Oh yeah, this guy, this little flagellate guy. I definitely see him. I definitely look at the sample. I was like, compared to yours, you had so many nematodes in your vermicompost. I feel like it had so much more. It just had more top level predators. Like here, I looked at other compost samples. Like this definitely had a good amount of fungi, at least the earlier sample did, and amoebas. But yeah, I don't see a lot of more top-low protozoas. Don't see a lot of nematodes. So maybe I would try a protozoan infusion and a nematode extract on here, and then like, take another sample then.
Sure, and you can assess over time too. Like I bet you if you were to look at your compost when it first came off, you probably saw a lot more bacterial feeding nematodes in there. And over
Yeah.
time, that population would shrink. And this again, you can still do this qualitatively. You would see these things and say, now 6 months have gone by, I'm seeing a lot less bacterial feeding nematodes, and the overall population of nematodes have gone down. And maybe you start seeing a switch now to more fungal feeding nematodes as
Mm.
being kind of prevalent in your compost, especially with the amount of fungi that you're growing in there. Yeah, you know what? It's a funny thing, vermicompost. If you, some of the best compost I've ever seen on the microscope has been vermicompost. Um, but I've also seen really horrible vermicompost too. It just, for me, sometimes vermicompost gets a lot of diversity because I'm also adding a huge diversity of inputs into there. All these different food scraps, and I pull in other things from the yard, and I put wood chip in there, and a whole bunch of very diverse materials. And it's over time, I'm adding more and more diversity. So I tend to see a lot of diversity in my worm bins.
I should have asked when Keisha's here. Like, there's the concept about the mother pile, which is the pile that it's basically like that you have sitting there, and you just keep adding diversity. You keep layering it with biology,
Yep.
just like it has the kind of the bank of biology that you could add to new piles. Like, Brian, what are you
Sure.
doing for that at your house?
Yeah, so and when I lived in Auburn, Northern California, I had a really nice mother pile. But when I moved up here to Newport, I didn't have a home to move into at first, so I had to give up all of that stuff. So I'm now creating a new mother pile. So I have a very small set of compost I've made from the multiple compost piles. But what you described, Raleigh, spot on. I always leave some amount of compost from that pile to add into my mother pile. So then I will use that to inoculate my new piles with. And the diversity over time continues to increase. So a mother pile is a great concept or practice to use. And we did have some questions come in around, I think, some scope. So Bruce
Oh
asked,
yeah,
Brian,
scope.
what model LW scope are you using in camera? It's the i4 Infinity that I'm using, and I have the BioVid camera that I'm using. It's a really sweet setup actually, for us I've used some other, what I would call this, the LW in my mind is what I would consider a mid-tier scope. There is the higher tier scopes, which would be like the Zeiss and the Olympus. Those are scopes that typically cost upwards of, $10,000 per scope. And they're great. Their optics are fantastic. In the mid-range, like Modic, LW Scientific, there's a number that kind of fit in that range, which is still using really good objectives. The bodies are well made, a little bit higher than like an AM scope or an OMAX kind of equation. And then, you get to the lower end scopes, just like an OMAX AM scope. Um, but as you can tell, Raleigh's using one of those right now, and the visualizations are perfectly fine. And I still use my OMAX when I need to go out to the field type of thing. So don't feel like you have to get a very high end one. But if you do want a little bit better optics, the LW for me has been a fantastic scope. I really like it.
Yeah, I'm pretty happy. Like the Omax seems to me like the entry level one, and then the LW would be the one that I would upgrade to get something like Brian's. Um, though it's definitely want to hop in. If I was ever doing like actual like serious lab work, I would probably end up. But I'm happy for this for just doing qualitative checks on
Sure.
compost. Though I definitely want to clean it. I'm at the point where I'm like, okay, I've had enough of these scratches on the lens. I want to get this
Right.
professionally clean because you have this annoying thing
Yeah,
that
and cleaning your scopes is important to do. Like Raleigh was saying, you can get them professionally cleaned. And there's a lot of, if you live next to a university or hospital, there's going to be somebody who does microscope cleaning because they get their scopes cleaned all the time. Usually it's a relatively inexpensive thing to do. For me, they'll usually come to your house and clean your scope for you. You can also learn how to clean it yourself. It's not horribly complex. Just got to make sure you got the right tools to be able to do it. All right, we had a couple more questions in here. Um, there was one about, let me go, sorry. Let's see.
A lot of questions in the chat versus the
Yeah,
Q&A.
looking there. So, chat. I think Stacy, I answered your question about the vermicompost. Yeah, I can see vermicompost being more diverse in the way that I do my vermicompost bins. There's a lot of vermicompost that's made it more like industrial scale, and the diversity of inputs is very narrow. In that case, then it's not going to be nearly as diverse. And this fits not just for vermicompost, it really for any compost. The more diversity of your input materials tends to equate to more diversity in your microbial life. They are just going to stimulate and feed different types of organisms. So that's the what I tend to see is more diverse inputs equals more diverse as far as the organisms I see. So Matthias asked a question. What what are criteria one should look at when buying a camera for a microscope? That's a great question. Cameras are a tough one. Um, the sensor size is important because the sensor is going to be, how much of a field of view do you have? Like when you're looking at Raleigh's right now, it's that square. You can see it's at 400x, but really the amount of field of view is going to be fairly narrow compared to my camera. My camera has a
Yeah.
very large sensor, so the field of view, in fact, the sensor on my camera shows a larger field of view than I see through my eyepieces. So a lot of times I'm looking through my camera, I'll see things on the edges that I cannot see through when looking through my eyepiece. So sensor size is something that's important. The other things I look for is what kind of connector do I have for my camera to be able to display. For those that are going to use USB to transfer the data, make sure it's USB 3.0. USB 2.0 is an older standard and it's much slower. You'll get a lot of not smooth video when it comes down to it. And it's really a video issue, not necessarily a picture problem. Um, but you also can also transfer other ways. A lot of the cameras will have HDMI, so you can go directly from your camera to a monitor or a TV, which is really a cool thing to do if you want to show with other people. HDMI is a fantastic addition to your camera. Some of them, like mine, have an SD card, so you can actually capture directly from the camera to an SD card. And it has a little interface when you hook up to a monitor to be able to have a mouse to click through. Mine also connects through the network. I have it right now hardwired through a Cat5 cable, and that's to my network here at home. And then my laptop is picking up the camera that way. Others, like this one, also has a Wi-Fi. So there's a Wi-Fi adapter I can put to it that can broadcast Wi-Fi. So those are some other considerations when you're looking at a camera to consider about how are you going to be able to observe or view that image that's coming off of there. All right. Let's see. We also some other questions here, Raleigh.
Well,
What are those dark pumpkin seed shapes?
let me try to find it. Yeah, I know what you're talking about. They're like the you're talking about these guys.
It could have,
I think it was the one that was more almost like football shaped. It's like they're larger
there's
than the
probably
normal fungal
spores.
spores. Yeah.
Probably fungal
I'll
spores. A lot
try to
of times a
find one.
football shape is going to be a fungal spore. Yeah. Um, and then Bruce said, if I understood you correctly, you said you can see more detail better looking through the eyepiece of a scope than through the video on 4K monitor. And yeah, that is the case. Even though I have a really nice camera, my camera is, one of the not the high end or the highest end of cameras, but it's what I consider to be kind of the upper end in that tier at least. And it does great videos and does great pictures, but I still see more fidelity, more clarity when I look through my eyepieces than I do with my camera. And as you go lower in the scale of cameras, you'll lose a little bit more of the fidelity, you lose a little bit more of that sharpness or crispness. Um, but it still may be good enough. You know what I mean? If you're just trying to take a picture of, let's say, a testing amoeba or, some fungal hyphae, it, the low end cameras can do a pretty good job or a decent job at that. When you start taking video, that's where a better camera tends to come into play. And then you're looking at what's the video format. Is it 1080p, 720p? Is it 4K? All those things. Like mine's a 4K, so I will display in true 4K. And in that sense, you're also looking for frame rate too. How many frames per second is the camera actually capturing? The higher the frame rate, usually the smoother the video option is concerned.
Yeah, also too, I'm here on Tube View. There's this thing called exposure time. Like if I can, it's brighter, but it also slows down the camera. So if I'm like,
Yeah,
if I pump that up, it'll be more choppy.
right.
But if I put it down, then it's a bit darker, but it's more smooth. So there's little
And
tricks.
Raleigh's using a tool called TubeView. It's
Yeah.
a camera software essentially. It's taking either picture or video formats, and then you can change the white balance, the color saturation. You can add grid lines on there. There's all kinds of really fancy things that you
Yeah.
can do to kind of improve
Ooh, the
that.
grid lines. I didn't even think about that. I gotta find that
Yeah.
feature.
We have
Yeah.
some questions in the Q&A as well. So there was one from Francis about, have you tried cooling the slide to slow down micro movement? Actually, to be honest with you, Francis, I have never done that. I'm assuming you probably could, although some of the microbes, may not slow their motility at all, or may just turn off completely. Um, but that's a good thing to do. We use on the other side of the house, though, we do use heat. So when I'm doing especially nematode identification, and you probably saw earlier when I was in my garden bed or the vermicompost, I had this one field of view that had three nematodes wiggling around like crazy on there. It'd be really hard to identify those. So what we do is we use something like this. And I use a torch. You can use a lighter if you want to as well, but this is like a kitchen torch. And you do is you basically heat up the slide from underneath, and you basically get into what we call a heat stun. So the heat will stun the nematode, and now you can observe it. It requires some practice because it's very easy to not apply enough heat, and then all of a sudden you haven't done anything, or you do too much heat and you actually kill the organisms, and then that becomes impossible to identify some in some cases. And also, if you don't want to spend too much time because you'll build up some soot on the underside of your slide, and then it fogs everything out. So, but we use heat to do some of that stunning.
Yeah, I've seen that work really
Um,
well.
uh, Levin asked a question about adding moisture to wake them back up. Yeah, there are times when I will have a sample that's very, very dry. And when I look at it, I see a lot of encysted organisms, so ciliates or the flagellates or the tested amoebas are in an encysted form. And then if I let them sit in a test tube for a couple days, then sometimes you'll see those decyst, and then you'll see those organisms start to wake up. So yeah, it can be interesting. And this is part of the microbe adventure stuff. It's pretty cool. Sometimes I'll make up a sample and then I'll let them sit for sometimes a week in my test tube, and I'll just come back later, a week later, and then take a look at it, see how things have changed. And it will. The microbial community will for sure change over that week's period of time frame. And so it's just being curious, I think, is the biggest thing. It's just let your curiosity go. And sometimes the curiosity didn't happen until I sat there and said, "Oh, that's been sitting there for a week. Let me go take a look at it." And then I'll look at it, and then I'm like, "Wow, it's very, very different." The biggest thing for me is just don't let those barriers get in front of you. If you have a curiosity about looking at something, definitely go look at it. We have another one from Gay that says, do you experience or do you have experience in epifluorescent light module? If so, is it useful for identifying mycorrhizae fungi? So I do have an epifluorescent scope. I rarely, rarely use it. Um, I can use it to identify mycorrhizae fungi, although it's a very difficult process. You have to get your working solution set correctly to get the fluorescence right. It's for me, for my clients, I actually charge quite a bit of money to do epi work, and so I don't get a lot of it, just because there's complexity involved with it. The materials you need are controlled substances, so you have to be set up with the lab. There's quite a bit of kind of regulatory stuff there. To be honest with you, my kind of move forward now with my mycorrhizae fungi is to be looking at doing more staining. I think that's a better way to go. I've talked a lot with Dr. Adam Cobb about, techniques and tools And he recommended there's kind of an ink-based staining that I haven't tried yet, but that's probably a way that I'm going to be looking to go. Just because you can really see them very sharp. Sometimes the epi, it's pretty hard to see the mycorrhizal fungi because they may not fluoresce very, very well. Whereas the staining, you tend to see it pretty sharp and pretty crisp.
Here's another question from
And then
Gail.
Gay had another question.
Yeah.
On Raleigh's screen, many test state are rather empty. Does that mean the sample needs more bacteria? Um, Not necessarily. It's like hermit crabs. There's a lot of times I'll find empty tests, just like if I go out on the beach, I'll find empty hermit shell crab shells, right? So it just means that the testing amoeba that was there had died. Um, Now, if I saw only empty tests, then that would give me an indicator. If I see some empty tests, but others that were full, then it's like, okay, then I know it's just kind of the regular mortality that's there.
Hey, Brian, real quick.
All right, and I see, yeah, go ahead, Brawley.
So I know like when you're doing quantitative, you're counting fungi, but one of the tough thing was it's like when you're looking at an aggregate that's just clumped with fungi, how would you accurately like add that to the quantitative analysis? Like you're looking at an aggregate and there's clearly like, all within this aggregate lots of fungi. Like how would you even
Sure.
factor that in?
Yeah, it's tough. What I intend to do is when I look at aggregates, I will get the aggregate right in the middle of my screen. I will usually jack up the light source to almost full brightness, as full as I can see it, to get enough light going through the aggregate itself. And then you use the fine focus knob very slowly and you just kind of like incrementally click down through. And then when I'm seeing things come in focus, that's when I'm taking counts of my fungi in there. Like you said, in some of the aggregates, they'll be loaded. Like you have an aggregate right there that's got fungi in it.
Yeah, it clearly it's like, oh, that's fungi. And so I wish I could
Yeah, go
focus.
go to the one that's onto the right hand side.
Oh,
That one
like,
right there. Yeah.
sorry.
So if you were to go through your focal plane, so watch Raleigh as he goes through that focal plane, you can see that fungal hyphae pop out, and then as he keeps to continue to focus, then it disappears.
Yeah.
And this is the important part of using your focal planes, because here you're able to look through that whole aggregate, and then you're able, and sometimes you'll see tested amoeba in there, you'll see flagellates in there, you'll see other organisms just besides that. So when you're looking through, just don't look at one focal plane. Go through the entire focal plane itself, and then look for organisms. So you can see when Raleigh gets to the right spot, that fungal hyphae, and Raleigh, if you want to put your cursor so you can show people.
Oh, this guy
So
or
yeah,
the big
exactly.
guy? Wow, probably.
Not the big guy, the one in the
Yeah.
aggregate itself.
Yeah,
Okay,
there's
so you
so
can see
many.
right there as Raleigh focused through. Yeah.
This is where I wish I had the LW because it'd probably be better at like finding, like going all the way through the aggregate and like kind of like being able to finally count and identify the fungi within
Well,
the
that's where
macro
sharpness
aggregate.
comes into play. Sure. You
Yeah.
get a little bit sharper of a look when you're going through there. But
Yeah.
Raleigh, look at your compost. This is fantastic compost. I'm seeing tons of testing amoeba. I've seen flagellate. You got good fungi in here. You might be a little bit low in the nematode, but you know what? At the end of the day, you could do a nematode extraction and kind of really understand what kind of nematode population you've got. I bet you probably are moving now more towards fungal feeders into your compost, plus probably some predatory nematodes as well.
Yeah, no, it's great. When I did the review on the other compost, it's pretty much the same thing. Lots of fungi, lots of amoeba. I saw like pretty much one nematode, but yeah, I would do an infusion, nematode infusion on this to try to bring that back up.
Yeah.
But yeah, it's just like an amoeba. I'm not sure if it's amoeba graveyard or amoeba like they had a
No,
giant party. No,
I see it, like right there. So if you see that the tested amoeba that's up on the kind of upper left hand side,
yeah,
that's an amoeba that's got, you can see there's
it's got
material
his,
that's inside of there.
yeah.
You have another one over to the right hand side. It's not quite empty. And sometimes you'll see also the testing amoeba are kind of cool when they get into like a threatened state or whatever else, they'll pull in material and plug up the front of their hole, right? So you can
Mm,
see usually like a plug of material in there as well.
nice.
And you got to realize too, testing amoeba, the amoeba itself is very translucent. Just like when you see naked amoeba. Sometimes naked amoeba are hard to see, um, just simply because of their transparency that's there. Like a testate amoeba is easy to see because the test usually will shadow very well. Where a naked amoeba, it's a little bit harder to see them.
When we do the next one, we got to have one sample that's just like either just a tilled the shit farm field or just the commercial compost, just so people could see the difference of what just
Yeah,
what people are normally dealing with in terms of soil quality. Like the
for
norm
sure.
is so low in comparison.
Yeah.
Wow.
Let's say one more question. We got one from Francis
Yeah.
here, which is, does using a measuring reticule on the eyepiece help in measuring quantitatively? So, a measuring reticule, and you can find them on slides too, but it puts grids in there, and it gives you some measurements as far as when you're looking through, so you can better assess, you know, lengths and diameters and things like that, especially when we're talking about fungi. They can help. I'm not a huge fan. They just kind of annoy me after a while. And I think really, if you're going to do a quantitative for a long period of time frame, you get just a skill to do it. You know, your brain and your eyes are good enough to be able to measure it. So maybe early on when you're learning how to do this, it can help out. But for me, for the long haul, it just becomes kind of annoying. And it just is a visual blocker for me. So. I don't use them in my practice anymore.
We,
All
I
right. Well, Raleigh,
think we
I think that
reached a good point.
I think we hit it. And just thank you everybody for showing up to our session today. Hopefully you guys were able to get something out of it. And hopefully get inspired to go out there. And if you have a microscope, just don't be afraid. Go out there and start taking samples and look at things. So important.
And also too, if you guys have questions like about like, hey, I might be interested in the microscopy course in Costa Rica, feel free to reach out to us, info@compost.academy. Love to answer your questions. Um, and of course, I've got a direct line that you can ask questions there too. It's just 925-330-0846. It's right on the website. But appreciate everybody joining. It was a super fun session, and next time we hope to see you there. We might do one more before the microscope course, but yeah, it was really fun having everybody on.
Yeah. Thanks everybody for showing up, and hopefully we'll see you in the classes.
All right, ciao everybody. Peace.
All right. Bye all.